when you start talking about plants and how do they move water through their body, you have to think about water potential concepts, which include both the idea of concentration ingredients, which is in here with diffusion. You know about diffusion processes in general. So we have to think about a concentration gradient, but we also have to think about a pressure gradient. So I'll incorporate both of those ideas today and introduce water potential to you. So I think we left off here before the long weekend looking at the idea of Fick's first law of diffusion. And the big picture idea here is that you have a concentration gradient, a difference in concentration from point A to point B, whether it be perfume molecules in a perfume bottle or water, thinking about the plant taking up water. Am I going to ask you to use this equation? No, but think about it, yes. So we're looking at a concentration gradient, and if you take this equation and do some derivation of it, you can come up with a way to calculate how long it takes for something to diffuse. And again, this is the equation that they use, distance squared, time equals distance squared times k and d, which are the diffusion coefficient is d, and then k is a constant that looks at the geometry of the system. And again, you can look this up. Am I going to ask you to use this equation? No, but distance squared is a key concept here. And so what this is saying is if your distance is very, very small, diffusion works pretty well. It's a pretty elegant process of moving solutes from point A to point B. So if you calculate how long does it take for a molecule, I think in your book they use a molecule of glucose. How long does it take to go from one end of a plant cell to the other end of the plant cell? About 2.5 seconds. So that's pretty good. When you're at the cellular level, diffusion works pretty well. If you start to just go a little bit bigger, like you say, what about a leaf? And I think in your book, they look at a corn leaf. And if you calculate how long does it take for like a molecule of glucose to get from one end of the leaf all the way to the other, 32 years. Well, obviously that's not cutting it. And so once the plant physiologists started doing the actual calculations, they're like, oh boy, diffusion can't be for more long distance transport. And when you think about giant sequoias and giant redwoods, where water has to move from the roots all the way to the leaves, that's a long distance. So they know that they have to think about some other processes. So they started to think about this, which is bulk flow. And bulk flow, again, this is the equation. They looked at the flow rate, and it's equal to pi times r to the fourth divided by 8 times n. And then you see there's a pressure gradient, delta P over delta X, a difference in pressure between point A and point B. So with this idea, you're looking at bulk flow is looking at when there's some kind of external force like gravity or pressure being applied to a system of some sort. With this equation, I'm not going to ask you to use it, but there's a couple of key points here. R is the radius of the system you're looking at. For our purposes, we're very interested in the xylem where water is moving. So what is the radius of the xylem cells? N is the viscosity of the liquid that you're dealing with. And then delta P over delta X is your pressure gradient. Going back to our radius, R to the fourth. That's a pretty sizable thing here. So this technically is the Poisson equation. I believe that's the right way to pronounce it. He was a French scientist, Poisson. He came up with this to describe how quickly substances move when there's some kind of gravity or pressure force being applied. And it does nicely work for long distance transport. It helped to explain how water can move. You know, they could, back then when you're trying to figure all this out, they could measure the time it takes for water to move from the roots to the leaves. And this helped to explain that quite nicely. Wasn't perfect, but it really did say, you know, they're like, okay, bulk flow seems to be playing a role, definitely. Now, That R to the fourth bulk flow is very sensitive to the radius of the system you're studying. Again, we're looking at xylem and how wide in diameter, or what's the radius of those cells. So there's two types of xylem. When you think about angiosperms, there's two types of xylem. You remember, one starts with a T and one starts with a V. Do you remember what those are? Vessels and tracheids. Do you remember which one's wider in diameter? 50-50 chance. Vessel. The way I remember is V goes up wide, the top of the V is wide in diameter. So that's how I remember vessels are the wider ones. So with vessels being a wider radius or diameter, their flow rate through those vessels is much quicker than tracheids. And, if you just do a simple thought experiment where you look at the radius, and if you take the radius and you say, well, if it's a radius of 1 to the fourth is 1. But then if you double the radius from 1 to 2, does anyone remember what 2 to the fourth is equal to? Math. Equal to 16. So a factor of 16, if you double the radius, the flow rate goes up by a factor of 16. That's a huge jump. So very sensitive to the radius. So tracheids versus vessels can make a big difference in terms of flow rate of water through that system. So now we're thinking about, they were doing these thought equations and then testing them out, taking actual measurements, trying to figure out how is water moving through, what forces are allowing for it to move through as quickly as we see when we take actual measurements. And so they did fairly quickly understand that it is what we call unidirectional flow through the plant body. When water moves through the plant body, it starts in the roots. So roots take up water from the soil, and then it gets dumped into the xylem, and then it courses through the plant body all the way to the leaves. And then eventually it will exit out of the leaves as water vapor. It shifts into the gaseous form and exits out of the leaf. So it doesn't go any other way. It doesn't go from leaves down to roots, or tree trunk down to roots, or it's one way. In through the roots, up through the plant, out through the stomata. So they figured that out, and then they started looking at how is it moving? Is it concentration differences? Is it pressure gradients? What's going on here? Well, they realized they had to start thinking about osmosis in order to more fully comprehend this. And osmosis is the movement of water. It's a two-part definition. Movement of water, when you're focusing on water movement. And then you're also thinking about a selectively permeable membrane. So this is the formal definition. Osmosis is the movement of water through a selectively permeable membrane. So I break it up into two parts here because you have to think about both of those carefully. So the classic example is to take a YouTube, which I have a hard time thinking about it like this. If I say YouTube to you, you probably think about YouTube videos. But this is YouTube. And what they've done is they've put a selectively permeable membrane right in the middle there. And then what they're trying to depict here is two different glucose solutions. One is more dilute on the left. The other one is more concentrated, more glucose. And then they say, okay, if we put, equal volumes of these two solutions down on YouTube and then wait for a little bit, what's going to happen? And I think if you think back to basic ideas of diffusion, you'd say, well, water is going to move. So it's differential, selectively permeable. Glucose can't move, but water can. So water is going to move from the membrane to the more concentrated solution to dilute it out. And that's, it's a fair way to think about it. And that's what they're trying to show here, that water is starting to move that membrane. Glucose molecules get left behind, and you can see that the water content or the solution, the volume increase over onto the right side of that YouTube. Okay, so really what we have going on here is both a concentration gradient affecting how water is moving here, but also a pressure gradient. So with this idea, initially, when you say, well, how is water going to move? You look at the concentration of the glucose molecules. Or you can also flip it if you want and look at the concentration of water molecules on either side of the membrane. But you're usually taught to look at the, in this case, the solute that's dissolved, the glucose. And that's fine. So you're looking at a concentration difference, a concentration gradient on either side of that membrane to get you to think about which way is water going to move. And based on that, you say, well, water is going to move to the more concentrated glucose solution. So it's going to shift over through the membrane over to the right. But I also said there's a pressure gradient impact here too, and that's often not really discussed so much. But in plants, we have to think about it because we have to think about the stem and the branches. It's a big long tube of tracheids and vessels, going up to the plant. So if you can imagine this being xylem vessels or tracheids, so you have water moving over to the right, and as you do that, you're starting to increase the pressure of the system, pressing back down on this membrane, right? So you do have pressure forces, standing water, or in this case, glucose solution. The higher it goes, the more that the water courses over there, the higher the pressure pushing back against that membrane, until it will reach a point where you're not going to get any more water movement because the pressure is too much, and you're not going to get anything more coming over. And so, yes, concentration kind of kicks off the idea, how is water going to move? But then immediately, as soon as water moves, you start to think about a pressure gradient that you have to factor into it. So they started to see this idea as they're, running tests and thought problems and doing the math and then taking some measurements of how water's moving through the plant. And they came up with, initially, they're like, well, we could calculate the chemical potential of water. That would be a way to try to describe or explain how water is moving through a plant. And if you've, I don't know if they teach about that, maybe in Chem 111 or 112, they might talk about chemical potential of water. And it's a micron symbol to denote that. But when you look up the definition, it's free energy per mole, in this case of water. You measure free energy by joules, that's the unit. So it's joules per mole is what we're thinking about here. And what they said was, well, you could say that osmosis is going to occur when there's a difference in chemical potential on either side of that membrane. And that's absolutely true. And then they say, well, you're going to get movement of water down a chemical potential gradient from higher to lower. And again, true. And they said, and all of that is influenced by concentration, either of the glucose molecules or how much water molecules are in the system, and then also by pressure. So they started looking at it from that angle, calculating chemical potential on either side of, in this case, the U-tube with that selectively permeable membrane. And here they did a thought question, like, okay, if we put a piston in that tube where the water is moving over, and we applied enough pressure to this to make sure that the water level doesn't rise, you can actually measure the pressure that's being generated as water's trying to get over there. And you can actually do that. And, they did these kinds of things where they were trying to imagine and test how things are, how osmosis is occurring. Well, they quickly realized, calculating the chemical potential of water, it's kind of a pain. It's a lot of work. So some plant physiologists said, why don't we switch it to water potential? Let's do that. And the way to denote water potential is the Greek letter psi, PSI. That looks like a funky Y. All right, so psi sub W is how we describe water potential. So they said, let's convert chemical potential over to water potential, because they found out water potential is easier to measure in real life situations than chemical potential. So the way to do that is to convert it. You can take the chemical potential of water and divide it by the partial mole volume of water. just take my word for it. And when you do that, you have a measurement of free energy per meters cubed. Okay, so this is another way to say per, do a negative superscript. That's denoting that the moles are in the, did I say meters? Moles are in the, no, meters, joules per meter, cubed. Okay. So this is to convince you that you can convert chemical potential to water potential. Again, I'm not going to ask you to spit this equation out. That's not what I'm going for here. What I'm trying to show you is that you can use water potential. Again, this is the previous equation I just showed you on the previous slide. Chemical potential of water divided by the partial mole volume of water, which is the chemical potential of joules per mole. Partial mole volume, if you're curious, that's meters cubed per mole. Okay, so follow this here. So joule per mole up here, and the chemical potential of water is joules per mole. When you look at that, another way to describe a joule is equal to a Newton times meter. This is just another way to describe it. So now, if we take Newton meter and put it in place of the joule, that's what's being shown down here. So you have the Newton meter instead of joule per mole divided by the partial mole volume, which is meters cubed per mole. When you cancel all that out and work it, you get, by canceling everything out, you get Newton per meter squared. Newton per meter squared. Well, Newton per meter squared, that's a unit of pressure. So what we've just done is converted chemical potential to water potential, and it's a unit of Pascal. Newton per meter squared is equal to a Pascal. It's abbreviated PA, and that is a unit of pressure. So now I've cut out a bunch of steps on derivation of all of this because I don't want to go on and on about that. But basically what this is showing in an abbreviated form is that they took the chemical potential, shifted it to water potential, and when you do that, you make measurements in pressure. You measure pressure. That's much easier. You can buy instruments that can measure pressure, really not too bad. And it's gotten less expensive over time. And so this is a more practical way to actually try to figure out how is water moving through the plant body. So plant physiologists are all about simplifying and making things easy so that we can measure things in real life. And so this is a good way to do it. Okay, so now we're going to kind of dump the chemical potential idea and focus in on water potential and think about how do we measure that? How do we think about it in terms of how water is moving? So now we need to think about a water potential gradient. And when you think about how water is going to move, we say that it moves from a high water potential to a lower water potential. And that's accurate. But when they started looking at water movement in the plant, they quickly realized that it's actually a negative value. It turns into a negative number when they take these measurements. So what you need to think about, if you look up the definition of tension, the definition of tension is negative pressure. Negative pressure. So when they're actually measuring things in the xylem and looking at how things are moving, they actually determine that you get negative values, which actually means you're dealing with negative pressure equals you're dealing with tension forces going on in the xylem. And so because you're on a negative scale, negative numbers and a negative scale, to rephrase this first bullet point, instead of saying from high water potential to lower water potential, a more accurate way would be to say that water moves from less negative water potential to more negative water potential. Now, if you've not thought about a negative scale in a while, you got to kind of slow down and give this some thought. I always had to do that, so we'll look at that again in a minute. They also decided it would make sense to measure all of this against a reference, so they call pure water at atmospheric pressure. They'll say that's water potential of 0. So you do need to know that, because this will come into play as we're thinking about some thought experiments on water potential and movement of water. So pure water, that means there's no minerals, no gases, nothing. It is pure water at atmospheric pressure. Then we say it's 0. The water potential is 0. Okay. Now, when they started collecting data on different plants, and they kept seeing over and over, there's a tension force in the xylem. So, a negative pressure. And so that's why they said, okay, really water's moving from less negative water potential to more negative. They came up with this simplified equation to describe most water movement in most plants. You have to add on a couple additional terms if you think about really tall plants. Then you have to think about gravity forces, and that's another term that often gets put on this equation. But for this class and for what we're doing, if you're dealing with typical plants up to about 5 to 10 meters high and down in that area, this is fine. That's another thing about plant physiologists. They call it good. They're like, yeah, that's good enough. So what is it? This means the water potential psi sub W. If you're going to actually solve it and work an equation, it's equal to psi sub P plus psi sub S. Psi sub P is what they call the pressure potential. And really, when you think about plants, you're thinking about water movement, so hydrostatic pressure, water pressure. Psi sub S is the osmotic potential. That is helping to describe how much solutes might be dissolved in that water. So you have to think about that. So again, this is going back to the idea of you have to think about a concentration gradient and a pressure gradient in order to determine water potential. So again, depending on how fancy you want to get, you might want to include a gravity impact. If it's a taller tree, psi sub M is matrix potential. Sometimes they'll add in psi sub M, especially in a soil system that's a little bit more complex. You could also incorporate it with the idea of water interacting with the xylem cell walls, but usually it's so minor they just omit it. So yeah, we don't need to worry about it. In the soil, though, if you're thinking about water protection in the soil, some people will say, yeah, maybe we should put psi sub N on there. So you just would put plus psi sub N and have that as an additional factor. But for our purposes, you need to know that top equation. That you do have to memorize. I will be asking you about it. Psi sub W is equal to psi sub P plus psi sub S. All right, so we have to think about a negative scale because water is going to move from less negative to more negative water potential. So here's your negative scale, how they usually draw it out on the board. And you can see that when you're in the negative scale, that as you approach 0 on the scale, you're becoming less negative. Those numbers are becoming less negative. As you get further away from 0 and go more negative, or further down the negative, -7, -8, -9, those are more negative values. Okay. I really, really prefer that when I ask you a question on the exam about water potential, you use the terms less negative, more negative, instead of higher and lower. Technically, you could describe it that way, but I have seen over and over through the years, students mix it up and flip it in their head because they're not used to thinking about a negative scale. So it's more, I think your brain would accept it better if you talk about less negative versus more negative values as you're describing what's happening. And then you're safe. you're more accurate at what you're describing. So this drawing here, somebody did a calculation where they said, we calculated this cell and it has an overall water potential of negative 0.25 pascals. So they factored in all the proteins and enzymes and DNA and molecules in there and magically came up with, we know it has a water potential of negative 0.25. And someone put, maybe it's a sucrose solution into the beaker. And they said, well, we calculated the water potential of that and it's negative 0.15. So now they're saying, okay, well, let's drop this cell into the beaker. Now, this is a cell that has a selective permeable membrane. So now we're going to think about osmosis. We're going to dump that cell in. You have a selectively permeable membrane. You have a difference in concentration on either side of that membrane. And in this case, all you really have to do is look at the overall water potential, and that will tell you how water moves. So now you say, okay, well, I know water moves from less negative to more negative. So then you look in the two parts of the system and you say, which number is less negative? So which of those? Is it 0.15 or 0.25 that's less negative? Which one's closer to 0? 0.15, right? So this one is less negative. So, okay, so that plant cell gets dropped in there and we say we know water moves from less negative water potential to more negative water potential. So that means water will rush into the cell across the cell membrane, which is selectively permeable. And you're correct. That's how in this case, that's how water will move. So water is going to rush into this living cell, a cell membrane, a cell wall. What force is going to start coming into play here? If you have a bunch of water rushing into the cell, what starts to happen to that cell in terms of what force is starting to really start to come into play? You put more and more water into a water balloon. Pressure, hydrostatic pressure starts to build up, build up, build up. So we're still thinking about concentration and pressure ideas. We're just describing it in terms of water potential and looking at your sites of P value, sites of S value. And so, you know, and we'll look at this a little bit more carefully where we actually know the values of sites of P and sites of S. This is kind of just a big picture idea where they just give you overall water potential. So let's look at this one. Here they're just showing if you have pure water, that's your reference. Pure water, open to air, open to atmospheric pressure, then your overall water potential is 0. Okay, so that's just a visual about the reference that they use. The figure B is looking at the idea of putting, sucrose. In this case, they put sucrose in there. So they put a 0.1 molar solution of sucrose. By the way, the capital M, the italicized capital M is molar. So that's moles per liter that you're thinking about, moles per liter. So 0.1 molar concentration of sucrose. Okay, so this is open to air, not closed off by any kind of membrane or anything like that. It's open to atmospheric pressure, so your pressure is 0. But someone did the calculation and said, if you have a 0.1 molar solution, that gives you a size of F value of negative 0.244 megapascals. And then they added those two up, of course, because the pressure value is 0, and then the overall potential of negative 0.244. Okay, so this is how they start doing these calculations. They look at what is the size of P, what is the size of S, add those together, and then that will give you your overall size of W. Okay. Well, let's do a thought problem where you have this condition. Anytime they say flaccid cell, if I say that too on a quiz or test, that means your size of P is equal to 0. That means it's kind of like wilted lettuce in a way. It's just, there's no pressure. There's not enough water in that cell to cause any outward pressure on the cell membrane or the cell wall. So your size of P value with a flaccid cell is 0. Someone did the calculation of how many solutes are in that plant cell, and they came up with negative 0.732. Add those together, and you get negative 0.732 megapascals for your overall water potential. Then here, saying, okay, go ahead and dump that. By the way, that beaker is back from this, the 0.14, which is negative 0.244 water potential. So that's right here, negative 0.244. So that's what the solution is. And then you dump. So then you go, okay, well, the cell has a water potential of negative 0.732. The beaker solution is negative 0.244. So then you have to ask yourself, which of those two numbers is less negative or closer to 0? And it's the 244 close to 0 and 0.7. So that means water is going to rush into that cell from the beaker when it gets dropped in. So again, water rushes into the intact cell. It's going to keep rushing and rushing and rushing until it starts to build up that size of P value. You're going to get hydrostatic pressure building up and all the water going in. And so if you have a lot of water moving in, you're going to get pretty decent hydrostatic pressure. A little bit of water moving in, pretty small. Okay. And here, what they did was they calculated the size of P. So you can manipulate the equation. Depends what the question is, what you want to know. If you have enough of the values, you can plug it in and rearrange and get your question answered. So here they were asking about what is the pressure of that cell when the experiment is complete. Kind of makes sense. Now you may be sitting there going, why should I care about this? Well, a plant really cares about its water potential. And when they started calculating water potential of different plants under different situations, they could see that they could come to some conclusions where they knew that certain values of the water potential could indicate either a plant is really healthy, it's well watered, it's getting its needs met, it's good to go. So if you're a farmer and you're growing plants for a livelihood, this is a big deal. Knowing if you irrigated enough is a big deal. I mean, your profit margin is on the line here. And so farmers care a lot about this. Anyone who's growing massive amounts of crops for their livelihood. So on the other hand, if you're, you can also get water potential values that are pretty negative. And that is telling you that the plant is undergoing water stress. So what you're seeing here on the x-axis is your water value. I don't know that there is such a thing as negative 0. I think that was a little bit of a whoops by the artist. So 0, okay, and then negative 1, negative 2, negative 3, negative 4. And then on the y-axis, you're seeing different processes that the plant is doing on a day-to-day basis. And you can see that as the water potential is getting more negative, you can actually start to see a decline. So the way to interpret this green bar is if it's nice and wide, that means things are good, everything's good. And then as it starts to taper down into a point, that means it's starting to taper off. So they're saying photosynthesis is definitely crashing at about a water potential of negative 2 megapascal. Protein synthesis is even more sensitive, as is cell wall synthesis, cell expansion. Those start to tank pretty quickly at modest water potential. So measuring the water potential of your plant, and there are, like I said, there's gadgets out there that do that. You can go out into your field and take measurements and measure it in megapascals. And then you can estimate how much you have to irrigate, how much you have to water those plants, because you don't want your plant crashing its photosynthesis. If you're trying to grow a plant, you want it to do a lot of photosynthesis so you can harvest it and sell it. So understanding water potential is a big deal. A couple of other things on this image are the top two things being listed out there: abscisic acid accumulation and solute accumulation. So they're actually showing by the way it's drawn here that you get an increase in abscisic acid with more and more negative water potential. You get an increase in the amount of solutes with more and more negative water potential. And so hold that thought. We're going to come back to that idea. So what this is really saying is the plant is deliberately doing this. Okay, the plant is deliberately cranking up abscisic acid. It's a hormone that the plant has. And when it senses it's getting droughty, I'm not getting enough water, one of the responses by the plant is to increase abscisic acid. The other thing that the plant is doing, it's sensing, I don't have much water available. It'll actually start to manufacture or synthesize more solute in its body. And that can actually be a good thing because it is playing with osmosis. If the plant starts to manufacture more and more solutes, if you start to increase more and more solutes, say you take and increase this sucrose solution instead of 0.1 molar, you put it to 0.4 molar. You increase the amount of sucrose in there. What does that do to your Cy sub S value, do you think? More solutes. What do you think happens to your Cy sub S? Does it get less negative or more negative? What do you think? 50-50. Less negative or more negative? What do you think? More solutes in the system. What do you think might happen to your Cy sub S? It becomes more negative. So that's a mantra I'd kind of like you to get in your head. More solutes, more negative Cy sub S. More solutes, more negative Cy sub S. And I can, if you're curious to know the actual laws of chemistry and physics on that, but basically in a nutshell, you're increasing the chaos of the system. And when you do that, it drives your Cy sub S more negative. Okay, so more solutes, more negative Cy sub S. So what the plant is actually doing is it's manipulating what its Cy sub S value is. Saying, oh yeah, I can't pull in any water from the soil. It's just really hard. There's not water. I can't get more water. It will make a bunch of solutes. And remember, when you have osmosis going on, you think about concentration differences. When you think about how's water going to move, you say, well, how much sucrose, or in the plant, it doesn't really make sucrose as the actual solute. It does some other things. But basically, it increases its solute concentration so that osmosis occurs. Water will keep moving into the plant from less solutes out in the soil, more in the plant body. And so it's forcing a situation where it keeps having positive water flow into its body until there's hopefully a rain event, and then it'll be good for a while. But if there's no rain event and it keeps going more and more dehydrated in the soil, eventually it can actually flip where water comes out of the plant body. And that's terrible. It's going to kill the plant. So I'm sure if you have had grown, plants in a pot and you come home from vacation and it's all wilty, part of the process, hopefully it's not happening, but it can get to the point where the plant is so wilted out that it actually lost water into the soil based on the laws of how water moves under those conditions. And then even if you water it, it can't bounce back. It just won't. So knowing and being able to measure the status of water in the plant, you do it using water potential measurements. So what you're going to do in class or in lab tomorrow, you're actually going to work with the white potato that's been bought from the store, and you're going to ask yourself, what is the water potential of that white potato tuber? And based on the experiment we provide, you'll actually be able to do that. And you'll use that water potential equation, psi sub W is equal to psi sub P plus psi sub S. And you'll be able to plug in your values that you're collecting the data, and you'll be able to actually calculate the water potential of that tuber. And it can vary. I mean, every tuber is going to be slightly different, and depending on how long it's been sitting in the bin at the supermarket, some are going to be more dehydrated than others. But basically, what you're going to be doing is soaking those tissues in different concentrations of sugar solution. And you're manipulating osmosis essentially. And you're asking, which way is water moving? Is it going into the potato tissue that I have soaking in there? Or is it coming out of the potato tissue? And then you'll weigh the tissue after it's sat in those solutions. Or you measure the length of the tissue, because you're going to take like a cork borer and cut a core tissue sample. And so you'll actually be able to measure water potential of those tubers that you choose to start working. And so this is a pretty straightforward experiment, but it's a good way to get you thinking about water potential, the psi sub P value, the psi sub S value, and then calculating your overall water potential of the potato. No, let me go. So here you're looking at kind of put it into perspective to things you might know about just from living your ordinary life. You know, a car tire is about 0.2 megapascals. 1 atmosphere is about 0.1 megapascal. So this is just trying to orient you to, if you haven't thought about pressure units and what kind of pressure are we looking at. So here you can see, again, why we care about water potential when you're trying to grow plants for a living. Here they're showing a pretty linear data set where you increase the water, you get an increase in the yield of the plant in terms of the overall biomass at the end of the growing season. And so irrigation is a huge deal when it comes to growing plants, and luckily nowadays there's better and better technology out there that's allowing growers to really precisely irrigate their crop plants. But being able to provide adequate water has big influence on crop yield. So it's a pretty important concept. And just a reminder, basic biology, hopefully they taught you about aquaporin proteins. That's how water's getting through the cell membrane. There's special channel proteins that allow for rapid movement of water through the cell membrane. And so what we're thinking about with an intact plant, rather than a potato tuber, when we think about movement through an intact plant, we have to think about water in the soil and then the roots. And we have to have a situation where the water potential is in favor of the plant being able to take up that water, dump it into the xylem, and then course it through the plant body all the way to the leaves. And so next time we're going to be thinking about, we're going to dive down into the soil and start there, because that's how the plant is getting access to water. We have to think about what is water doing in the soil, and then how does the plant create a situation, so to speak, where it can take up that water from the soil environment and use it. The last couple of slides are looking at a thought question on dropping in a cell into the beaker again. So if it's flaccid, what is your psi sub P? Equal to what? 0. So if I say flaccid, just psi sub P is 0. Someone did the calculation of the cell, and its size of S value, its solute potential, osmotic potential, was negative 0.85. So overall, the water potential is negative 0.85. Then somebody did the calculation of the sucrose concentration. It's open to air, so there's no pressure. And then they said there's enough sucrose in there to create an osmotic potential of negative 0.25. So at the start of the experiment, before the cell gets dropped in, you have negative 0.25 in the beaker, and you have negative 0.85 in the cell. So you dump that cell in, well, less negative to more negative. That's how water is going to move. Well, price of W of 0.25 versus 0.85. 0.25 is less negative, it's closer to 0. So water is going to move from the beaker into the cell. So that's what they're showing. They drop the cell in and water's rushing into the cell. Okay, so this is what we just had in the previous image. Now, after the experiment, after they dropped that cell in, somebody came along and measured when water rushed into that cell, it started to generate a hydrostatic pressure. So your size of p-value immediately came into play, and they calculated it was 0.45 megapascals. And they said, with the water moving in, you actually see a drop in the osmotic from negative 0.8, whereas at the start of the experiment, it was negative 0.85. So essentially what this is showing in numbers is that when water rushed into that cell, it diluted out the solutes that were in that cell. So if you make a more dilute solution, you're going to make your size of S less negative. That's what you see here. It goes from negative 0.85, which is more negative. Negative 0.8 is less negative. So then the overall water potential, when you add those two together, is negative 0.35. The beaker, somebody came along and said, well, it's open to air, so your size of P is 0. You lost water out of that solution, but the sucrose stayed behind. So you made that solution more concentrated, more solutes, more negative size of S. So that's what you're seeing here. The size of S went from negative 0.25 to a more negative 0.35. You add those two together, and you end up with a size of W of negative 0.35. So at the end of the experiment, at equilibrium, you should have identical water potential on either side of that membrane, which in this case is the beaker and inside the cell. So when we say at equilibrium, what we're saying is there's no net movement of water. There's like one-to-one exchange of water. You can still have that happening, like a molecule of water and a molecule of water, but you're not going to have a net inrush. When we say equilibrium, that's no more net movement of water. So please look this over. You're going to be thinking about these ideas in lab tomorrow. On Friday's lecture, I'm going to ask you some questions. And as a group here, we're going to work through some of these ideas to get you thinking about it. And I want to be here so I can answer questions. I don't want to assign it as homework. We'll just do it here in person so we can work through it together. Okay, so think this stuff over and have fun in lab, and we'll talk a little bit more about it in lecture on Friday.